Hi, my name is Susan Weekly. I'm the Campus Biosafety Officer, and you're going to learn bloodborne pathogens training. The purpose of the training is to minimize or eliminate the risk of personnel exposure in the workplace. That's EH&S's primary responsibility, and that's what you'll learn today. Also, we want to inform you of any potential exposure that you'll have in the workplace and protect the health and safety not only of you as you're here in our facilities, but of the community as a whole. The issue with infectious agents, if you end up working with infectious agents that are communicable, is that you're putting yourself potentially at risk if there's an exposure, other people in the lab, possibly your families if there was to be an outbreak. So we want to make sure that not only your health and safety is protected, but the community is also. Some basic information about bloodborne pathogens. We want to make sure that you have an understanding of infectious agents, biosafety levels, your requirements for personal protective equipment, what to do in case of emergency, and possible exposure.
The most optimistic projection USAMRIID is willing to make for the spread of the virus is this: 24 hours, 36 hours, 48 hours. The containment procedure must be viewed objectively. Be compassionate, but be compassionate globally. Thank you.
All right, please, please. The President's ETA from the East Asia Economic Summit is about 20 hours from now. He wants a recommendation by then from this group. Now, as I understand it, you want to firebomb the town of Cedar Creek, California, population 2,600, with something called a fuel-air bomb, the most powerful non-nuclear weapon in our arsenal. The way it works, it explodes, sucks in all available oxygen to the core, vaporizes everything within a mile of ground zero, men, women, children, and one airborne virus. Destruction complete, case closed, crisis over. This Constitution of the United States, I've read it cover to cover. I don't find anything in it about vaporizing 2,600 American citizens. But it does say several times that no person shall be deprived of life, liberty, or property without due process. So, a couple things before a clean sweep is even considered. One, unanimous, unwavering support for the President on this one. And I mean public. You're going to stand there shoulder to shoulder with him. He goes down, you go down. And the second thing is, I want an army of experts citing hundreds and thousands of lab experiments, telling any idiot with a camera that there was no other way. You got that? No member of this government is going to go sneaking off to the Washington Post telling them how they were the sole voice of opposition. If there is a voice of opposition out there, I want him in here, now. Those are the citizens of Cedar Creek. Go on, look at them. These are not statistics, ladies and gentlemen. They're flesh and blood. And I want you to burn those into your memories, because those images should haunt us till the day we die.
This is a required annual training for anyone that's using human or primate cells, tissues, or body fluids, any infectious agents at level 2 or higher, or working with level 3 pathogens. Also, it's required for anyone that's designated as a first responder or that's required to do first aid as a part of their position. There are lots of biohazardous materials that we use on campus. We want you to be aware of what they are, so if you encounter them. We're using things like viruses, bacteria, fungi, chlamydia, rickettsia, recombinant DNA, transgenic plants and animals, human or primate cells, cell culture, tissues, or body fluids. We have a lot of labs that are doing that kind of research. Human brain tissue from demented patients that have a risk of having prions associated with them. Animals that are potential reservoirs of zoonotic diseases. So if you're going out in the field and doing capture of wild animals, we want to make sure that you understand the risks of diseases that the animals could have that could be passed on to you, such as field mice could have hantavirus, bats could have rabies, squirrels could have plague. So we want to make sure that you understand risk if you're doing field research. Also, we have people who are specifically putting infectious agents into animals trying to treat diseases, like HIV, obviously, cancer, and viral vectors. We also have a list of materials that can be used for bioterrorist potential that's available on our website that you can reference. Things like anthrax would need to be registered with Centers for Disease Control and Prevention, and we'd have to get a registration for those materials. Other things like botulinum toxin, tetrodotoxin. So if you intend to use any of these in your research, you'll need to get a registration through H&S. We also have some basic definitions you need to understand. Blood, human or primate blood or any of its components, please treat them as if it's high risk, as if it's infectious. Bloodborne pathogen, any microorganism that can be present in blood that can cause disease. The big three specifically called out in the standard are hepatitis B, hepatitis C, and HIV. We also have other bloodborne pathogens that can be used, but those are the three that we're going to specifically address in this training because it's part of the requirements that we do so. Engineered sharps injury protection. If you're going to be doing any extraction of blood in patients, you'll be required to use needleless devices or sharps with engineering controls in order to make sure that you don't get punctured from the time a needle leaves a patient's arm until it's disposed of in a sharps container. The other times that we may require sharps with engineering controls would be if you're doing injection of infectious agents into animals. We want to make sure that we limit any exposure potential to sharps, so we may ask that you use a different kind of device for that kind of research. If you have any questions about that, please contact me and we'll discuss it.